Good afternoon everyone. Today we are going to discuss the topic enzymes. If I'll talk about the nature of the enzymes, that enzymes are proteinaceous in nature. Okay, so basically I can say mostly enzymes are made up of proteins. But yes, there is an exception also, ribozyme. Ribozyme, it is RNA. It is not proteinaceous in nature. Ribozyme is RNA that works as an enzyme. Okay, so no doubt, mostly enzymes are proteinaceous in nature. That is, they are made up with the help of amino acid. But a few exceptions are there. That is, there is a RNA that can work as an enzyme, and that RNA that works as an enzyme, that is known as ribozyme. Okay, now no doubt mostly enzymes are proteinaceous. Now you have read about the different structure in case of a protein: primary structure, secondary structure, tertiary structure, quaternary structure of proteins. This you have done, right? So basically enzymes, they possess tertiary structure. Mainly I am saying, okay, what happens in primary structure? Actually, the amino acids are linked. This is primary structure. In secondary structure, this chain will be coiled in the form of a helix. This is secondary structure. And in tertiary structure, what will happen? This chain will coil upon itself. There will be so many foldings like this. Just see here how the enzymes look like in the tertiary structure. Can you just imagine? The chain, it is coiled upon itself only. Now, due to this folding, at a few places there are present depressions, cracks, crevices. Okay, like this. Here you can see. Here. Am I clear? Or let me make it even simpler. Like this. Is it fine? Here the chain is coiled like this. So like this. So many cracks are there, so many crevices are there. But here is an important point. One of the crevices is known as active site. Active site of the enzyme. Okay. Basically what happens at this site? There is abundance of amino group, carboxylic acid group, OH group. So basically this is working as an active site of this enzyme. Now what do you mean by the active site of the enzyme? Something is clear from the name, active site, the functional part of the enzyme. Okay, first of all let us talk about what is the function of enzyme. Right now we have discussed only this thing, that enzymes are made up of proteins. Clear? Now let us talk about what is the function of enzymes. Enzymes catalyze the reaction. They are biocatalysts. Enzymes are never used in the reaction. Okay, they take part in the reaction, they increase the speed of the reaction, but they are not consumed. After the reaction, we get back the enzyme again. Let me give the example. Suppose here I am having the enzyme. This is the active site. Okay, now there will be a substrate. Substrate means the chemical compound on which the enzyme will work. Suppose here I am having the substrate. Now there is a specificity. The shape of the substrate will resemble the shape of this active site. Like this you can understand what, like suppose Suppose here I am having this substrate. Now this can be fitted into this. So this is enzyme and this is substrate. Now what will happen? This substrate will get fit into the active site. Again I am saying the shape of this active site will be specific to the shape of the substrate. Suppose if the shape is like this. Just see here. If the shape is like this. Now this enzyme is having the active site like this. Now this substrate cannot fit here. There will be another substrate that can be catalyzed by this enzyme. So enzymes are specific in nature. Am I clear? So here a new substrate has to be made that will be having the similar structure like this. I can make like this. Like this. Here you can draw anything, but here the shape should be similar. So now what will happen? This substrate will enter in the active site. Okay, now let me draw. Now the substrate is here. Fine? So, now what will happen? The substrate will enter into the active site and now the enzyme will bind strongly with this substrate and it results in the formation of enzyme substrate complex. This is short-lived. What the enzyme will do, it will now start breaking and making the bonds. Anything can happen in the case of a chemical reaction. There could be breakage of some bonds and there could be formation of some bonds. So this enzyme will catalyze the breaking the bonds and the formation of new bonds in this substrate. Now what will happen? This substrate will change into product. Maybe one product, maybe two product. Let me draw two products. Now what will happen? This substrate will get changed into product. Now just see carefully what is happening. Suppose I have drawn two products. Suppose. Okay. So now this is enzyme product complex. Again, what will happen after some time? These two will get separate. Means enzyme will be separated out. And the products, they will be separated out like this. Am I clear? So that's what I told you. We got back the enzyme once again. Enzymes are taking part. They are increasing the rate of the reaction. They are increasing the speed of the reaction, but they are not being used. We got the enzyme back again. And the substrate has been changed into the product. Is that fine? So what enzymes are doing? Firstly, enzyme is there. It will bind with the substrate, and where the substrate will bind at the active site of the enzyme. Now it results in the formation of enzyme substrate complex. Now after that there is formation of enzyme product complex, and after that the enzyme and the product will get separated. That's what I told you. Enzymes are not used in the reaction. They are just catalyzing the reaction. Now let me give you one example with the help of which you can easily understand. In a biochemical reaction, the rate can be increased to what extent with the help of the enzymes. Okay, let us talk about an example for this. So there is a reaction that is going on in our body. CO2 reacts, CO2, that is carbon dioxide, reacts with water and it results in the formation of carbonic acid. Now suppose there is not involvement of any enzyme. So what will happen? This reaction results in the formation of H2CO3. Okay, now in the absence of enzyme, in 1 hour, only 200 molecules of H2CO3 are produced. But if there will be presence of enzyme, if enzyme will be present, that is carbonic anhydrase, then what will happen? This reaction will catalyze, in 1 second I'm just saying now, 6 lakh molecule of H2CO3. Now just compare. In the absence of enzyme, only 200 molecules of H2CO3 were produced in 1 hour. But if carbonic anhydrase is present, then in only 1 second, 6 lakh molecules are produced. It means the reaction is undergoing 10 million times faster. Clear? So just understand, the enzymes, they are increasing the speed of reaction to such extent. Am I clear?